at the University of Washington with an interdisciplinary team, and we are studying the biosignatures, cellular and proteomic, of extremophiles subject to cold and highly saline conditions. And I'm going to be showing some of our first results in this ongoing project. So first, we want to think that most of the moons and planets of astrobiological interest in the solar system have surfaces with temperatures well below the limits of life. And even though it's possible that organisms may be living below in deeper, warmer environments, these organisms may be transported towards the surface, which is where we are going to be looking for the biosignatures. So this raises the question of what happens to an organism when you take it away from its habitable region and you move it in colder and very likely saltier conditions. And to answer this question, we took a psychrophilic organism and we incubated it into salinity conditions far from its growth range. So what you are seeing here in this temperature salinity graph, you have the temperature on the vertical axis, salt concentration on the horizontal axis, and this square, this rectangle, is showing you the limits at which our organism can grow. So you can see it has a nice range of temperature going down to -12, but a rather limited range in salinities. So what we did is we incubated it at temperatures in the lower range of its temperature growth range, and salinities, a couple of salinities, both optimal salinities and higher salinities than what we know it can tolerate. And after incubating it, we measured the activity by incorporation of a radioactively labeled amino acid. We measured the viability, that is, we took the organisms after the incubation and we returned them to their optimal conditions to estimate the number of viable cells that were remaining. We observed the cells at the microscope with DAPI stain, and we did proteomics with tandem mass spectrometry. So I'm going to walk you through the rationale of what temperatures and salinities did we choose, moving into the methods and some of the results. So you see here a simplified diagram of the phases of seawater. When it's freezing, you have, again, temperature and salt concentration on the bottom. And basically, this diagram is telling you what phases are going to be stable at which temperatures and salinities. So you have liquid water in the warmer areas, frozen ice in the cold, and salt at higher salt concentrations. I also want to note that sometimes you can get solutions out of equilibrium. So you can get supercooled solutions, which are common for all of us studying in the laboratory, this type of systems. So you have liquid water at lower temperatures when it should be frozen. And an important thing in this diagram is I want to signal you to the brine equilibrium line. So here, what you are going to be seeing is, as this ice is freezing, the liquid inclusions within the ice are going to have a salinity that is dictated by this line. So for instance, if you are having ice at -10, the brine inside the ice is going to have a salinity or a salt concentration with an ionic strength of 3.2 approximately. And this is important because if you are an organism living within the ice, this means that as you lower the temperature, you are simultaneously increasing the salinity. So the organisms in the ice are experiencing these two stressors at the same time. So here it is in the phase diagram. You have here located the square with the rectangle with the growth conditions of our model organism, which you may have heard in other talks, Colwellia Psychrerythraea strain 34H. And when we zoom here, you can see the conditions that we used in the experiment. So we chose two temperatures, both in the growth range of Colwellia, -5 and -10, and we choose the following salinities. We chose a seawater, supercooled seawater, and we chose the brine salinity expected at the -5 temperature. And for -10, we did the same thing, seawater, supercooled, and we chose the -10 brine salinity. So the ionic strengths of those solutions would be 0.7 for the seawater, 1.8 for the brine equilibrium at -5, and 3.2 for the brine equilibrium at -10, and I have there the concentrations in parts per thousand for comparison. So let's take a look at what we did with the organisms themselves. We grew them in their optimal media at 0 degrees, and then washed the cells, transferred them to the high salinity media. The solutions were prepared following the expected concentrations for seawater ions at any given temperature. And then we amended the solutions with either a high or a low concentration of nutrients. We distributed these resuspended cells into vials to be into two sets of vials, one of them for the activity measurements, and another one for, at the same time, we took measurements for viability, cell number, and proteomics. Because we wanted to do a study of how this evolved through time, we took triplicate samples for the activity in a shorter time frame from 2 hours to 12, 24 hours, and 1 month. And we took samples for cell numbers and viability at 1 month and 4 months. After 4 months, we wanted to see how the organisms had been evolving their proteome after being subject to these different conditions of temperature, salinity, and nutrients. So let's take a look at what the organisms were doing, starting with the activity. So this graph here, you can see the activity in the vertical axis and time from 0 to 24 hours after the incubation. The vertical lines show you the activity for the two supercooled solutions. In green, the -5, in orange, the -10. And you can see that the brine activity, the incorporation of the radioactively labeled amino acid, is basically here in the bottom. You can barely see it above the activity of the kill controls. So as soon as these organisms experience a higher salinity, there is a drastic drop in their activity, in their metabolic activity. And after 1 month, if you compare the activity here in the vertical axis with the viability of the cells in a logarithmic scale in the horizontal axis, you can see that the populations radically diverge. So you still have higher activity after 1 month in the supercooled samples, lower activity in the brines, and what is interesting is the changes in viability. So for -5 degrees, you have a higher viability. So these cells are really happy. They are active and they are viable. When you increase the salinity at -5, the activity drops, but the viability is still relatively high. But for the supercooled -10 seawater, the viability has dropped. So after a month of incubation, even though they were picking up the amino acid at some point, they lost the ability to reproduce. And the cells that were in the -10 brine, they have no recognizable activity or viability after 1 month. So this is really stressful for them. And you can see that the salt is driving the difference in the activity and the temperature, the difference in the viability of the organism. So you have here in this area growing cells, here cells that are with low activity but still viable, cells that are active but not viable, and here in this condition, which is when most of them are non-viable and non-active. So let's take the cell numbers. Here you have the cells and the viability, and I'm going to be showing 2 treatments. The light colors are going to be without nutrients, and the dark colors with nutrients, and these triangles here show you the initial conditions. So these were the cells, how the state of the cells when we started incubation. And after 1 month, in the -5 supercooled seawater, most cells are similar to how they started, except that if they don't have very many nutrients, the number of cells decreases. And this outlier here is interesting because this sample was frozen. So the organisms here were actually experiencing not the seawater salinity, but the brine salinity at the -5 temperature. And that had an effect in the viability. So let's take a look at the brine samples. How did they do? The brine samples effectively with the brine, the viability decreased, and it was more drastic effect when there were no nutrients available for the organism. At -10, there was a bigger loss in the viability, and the effect of the nutrients was even higher. So cells really need the presence of nutrients to be able to survive incubations in these more extreme conditions. Here we had a lot of frozen samples, which we think were driving even further down the viability of the organisms, because these samples were experiencing the brine conditions at -10, and those were really extreme for the organism. At -10, the viability had dropped to 0, but what is interesting for us is that the number of cells was preserved. So these cells that were suddenly exposed to a very high salinity, they couldn't maintain their activity, neither their viability, but the cells remained. And we think that this may be important when you think in the context of finding biosignatures somewhere else. So what were these cells doing? For that, we're going to be looking at the proteome of these cells. I'm going to be showing 2 plots. The first one is an MDS plot in which each one of the dots is comparing the proteome of that sample with the proteome of all of the other samples. The second type of plots I will be showing are string networks in which each one of the dots is a protein, and what you are going to be seeing is how the proteins are related to other proteins, where they are, and how are they related in the metabolic pathways. So when you look at the proteome, it's nice because the treatments are separating. We have here the -5 with and without nutrients, the supercooled. This is the brine. Here up you have the supercooled -10, and here are all the -10 brine cells that were not doing very well. So let's see, what do the cells do when you increase the salinity? When you go from supercooled seawater to the increased salinity, the supercooled seawater cells are doing the normal metabolic paths of the cell. But when you increase the salinity, they start pressing all of the stress response. They become motile. They start assembling clusters of iron, which we think can help with the stress of the oxidative stress, and they start repairing their DNA. So they are stressed and they are responding to it. These two treatments had nutrients. In the absence of nutrients, what are the cells doing? So they are looking for metals, we think specifically iron, and they start expressing an interesting protein, which is called aconitase. And we think it's interesting because this protein has a remarkable stability at high salinities, according to the literature, and also can take a dual role depending on the presence or absence of iron. So we see that the cells are looking for iron and that they are overexpressing a protein that has something to do with, or that uses iron and can have different roles depending on the presence of iron. Now let's see how the metabolic networks looked. So here you have the -5 supercooled brines, and you will see how they compare with when the salinity is increased. So here is supercooled -5, and here is the increase in salinity. As soon as the salinity increases, a cell that initially had a lot of metabolic networks active and working simplifies its proteomic network to just focuses on finding metals and become mobile. When you do not have nutrients, the networks are simpler, and they are again focusing on acquiring iron and expressing the aconitase protein. When we compare these -5 brines with the more extreme -10 brines, we see that the proteome has simplified into making proteins that are going to stabilize the DNA under extreme conditions. So these cells are really stressed, just looking to maintain their DNA. So to conclude, what do we see when we expose a psychrophile to extreme salt conditions? They decrease the activity, they reduce the metabolic pathways, focus on iron acquisition and motility. In the -5 brine, the nutrients help it be viable. And in the -10 brine, they lose all the viability, and the proteome just simplifies to take care of the DNA and make it more stable. If you think about the biosignatures, we think that the aconitase may be a good candidate because it's overregulated in these cells exposed to the brines, and it's also very stable at high salinities. And the other aspect that we found interesting is that the cells are preserving their shape and DNA even though they are exposed to very high salinity conditions. And this is even after 4 months of incubation. So I want to thank everybody in the team, including Miranda Much, that is just joined our team 2 weeks ago to help us process all the proteomic data, and our funding agencies. Thank you.
Right, do we have any very quick question?
Hello, Justin Lawrence, Georgia Tech. That was a super interesting talk. I'll say the second question, which is much longer. The first question was, you mentioned the supercooled brines. How do you hold a seawater salinity sample at -5 for a month?
How do we hold it liquid? Actually, that's very common. It's the opposite state is how do we make them freeze?
Okay.
So because these are Eppendorf tubes and we are used very clean water to make our samples, the water just is in supercooled state because it doesn't have anything that nucleates the ice. So for us, the problem is actually freezing the samples.
So the Colwellia doesn't nucleate?
Colwellia doesn't seem to nucleate the ice. We thought it could help, but it doesn't have an effect. The only thing we have found is scratching the tubes. That does help with freezing.
Thank you.
Thanks.
Right, thanks again for our speaker.